Bye.
Hello, good evening, everybody. I hope all of you are doing fine and a very happy Independence Day to everyone.
Happy Independence Day, ma'am.
Okay,
Happy Independence Day, ma'am.
happy Independence Day, everybody. So, we'll just wait for 2 to 3 minutes to let other peoples also join the class and then we'll start the class. म्यूट कर दिया मैंने मुझे डर लग रहा है इसे बंद करूंगी तो चला जाएगा ना नेट
खुला रहने वाईफाई नहीं जाता है नीचे से चला जाता है।
हाँ तो अभी कुछ खड़खड़ करने वाले हो क्या किचन में
दाल में खा रहा है।
ओह
आ गई।
बंद कर लेते हैं
तो लिखकर तो कॉल करें।
हम्म Okay, then I think we should start the class. Probably the students are celebrating Independence Day today. So, anyways, okay, just a minute. So, is my screen visible to everybody? Is my screen visible? The presentation is visible or not? Uh, is my screen visible?
Yes, ma'am.
Yes, ma'am.
Okay, okay. Fine. So, today we'll be just doing some questions about the week 4, that is basics of cloning. So in week 4 we have studied about iPhone. Sorry. Yeah, sorry for the disturbance. So in week 4 we have studied in lecture 1 first the molecular tools for cloning. So in this we have studied about the enzymes which are used for molecular cloning. So we have studied about the restriction enzymes, ligase, alkaline phosphatase, linker molecules, adapter molecules. So in detail we have discussed about the restriction enzymes. So in restriction enzyme, what is restriction methylase system, how we do the nomenclature, what are the different types of restriction enzymes, what are the properties of type 2 restriction enzymes, and how to set a restriction enzyme reaction. So today, apart from doing the questions, what I have decided is that what we'll do, that because I do cloning in my lab on a routine basis, so I will give you an example of how to set up a digestion reaction and how to set up a ligation reaction. So sir has already explained you how to set up a digestion and a ligation reaction in his classes. So we'll just have a quick recap of it, and then I'll just do the calculations, and so that you can have a hands-on experience of how to set a reaction. Fine. And second thing is that, again, like we did in the last class, that whatever topics that you could not understand, we'll be discussing those topics also. And so likewise we'll go. Okay, so let's start with the questions first. Now students are entering. Fine. Okay, so the first question is, the genome of the bacteria is not cleaved by its own restriction enzymes because A, restriction enzymes do not have the specificity of its own DNA. The DNA of the bacteria is non methylated. The restriction enzyme sites are not present in the bacterial DNA. Or D, the DNA of the bacteria is methylated. So, what is the correct answer? What is the correct answer of question number 1? Anybody who wants to try? Why the genome of the bacteria is not cleaved by its own restriction enzymes? Nobody knows the answer? I want some participation from the class. Nobody has any answer? What should be the answer of
Ma'am,
this
is it
question?
A?
No, it's not A. So,
Ma'am, is it D? Ma'am, C.
the answer is D. So, just one quick question. Could you understand the restriction methylase system that was in the lecture 1? Or were there some confusions, because this question is based on restriction methylase system? So could you understand the restriction methylase system? I don't think so. That if you have problem, then I can explain you, because this question is related to that only. It's the very first
Ma'am,
thing.
can you revise it just?
Okay, so I'll just revise it. It was in lecture 1. Yeah. So So now just a basic concept of restriction methylase system. So what are bacteriophage? Do you know what are bacteriophage? Bacteriophage are? Do you know what are bacteriophage?
Viruses which
Nobody.
bacteria.
They
Huh?
cause diseases in virus.
No.
Ma'am, viruses which cause disease in bacteria. Infect
Yes.
bacteria.
Yes. So bacteriophage. So phage means that, Aishwarya, you are right. Bacteriophage are the virus that infects bacteria. So phage means virus and bacterio means that virus that is infecting bacteria. So basically, if we say that bacteriophage are the virus that infects the bacteria. Now what happens, that because bacteria are getting affected by bacteriophage, so bacteria have made a system so that they can protect themselves from bacteriophage. Okay, and that protection is actually this restriction methylase system. So in nature, these restriction enzymes occur, but they don't occur because these enzymes don't know that humans will do cloning, so there's a need of restriction enzymes, so we have to produce. No. So basically bacteria have these restriction enzymes so that whenever bacteriophage is attacking a bacteria, so its DNA is cleaved by these restriction enzymes. Okay, so these restriction enzymes are basically a defense mechanism by the bacteria, by which it cleaves the external DNA which is coming to its uh, territory, which is coming to its genome. Now these restriction enzymes will cleave the external DNA, but the sites of the restriction enzymes, it can be present in its own DNA also. Suppose if I am taking this EcoR1 GAAT-TC. Now this GAATC will be cleaving the DNA whenever this sequence is present in the DNA. Now this sequence can be present in bacteria's genome also. So bacteria has made a system by which it protects its own genome, but it cleaves the genome of other foreign organisms. So bacteria has a system to differentiate between self and non-self, that is self and foreign. So restriction means that it is cleaving the genome of the foreign DNA, and methylase means that it is methylating its own DNA, so that now once the DNA is methylated, now the restriction enzyme is not able to cleave it. Okay, so restriction methylase system is a kind of a defense mechanism by bacteria, by which restriction means it cleaves the DNA of foreign genome, and methylase means that it methylates its own genome, so that it is able to recognize self and non-self, and therefore it is not going to cleave its own DNA. So just like in humans we have autoimmune diseases, autoimmunity, where what happens that the cell starts attacking the cells, that autophagy or the death of our own cells starts happening. So just like humans have a system to differentiate between self and non-self, likewise bacteria also have a system to differentiate between foreign and the self DNA. So self DNA would be methylated and the foreign DNA would be cut by the restriction enzyme. Is it clear to everybody?
Ma'am, I couldn't understood it. Will you please repeat it again?
Yeah, so you see, um so there is a bacteriophage, okay, or any foreign DNA, which is, this is a bacteria. Fine. Now bacteria has its own genome. Suppose a bacteriophage, it's not a very good bacteriophage, but something like this, bacteriophage looks like this. So this bacteriophage is attacking the bacteria. Now this bacteriophage has released its DNA. Now, this bacteriophage wants to take over the machinery of the bacteria, so that the bacteria will lyse and bacteriophage is able to produce. Because that's how viruses produce themselves. Now, this bacteria is also very clever. What it will do, bacteria has its restriction enzymes. So now these restriction enzymes will go, suppose bacteria released a restriction enzyme, EcoR1. Now, wherever there is this site in the foreign DNA, this EcoR1 will cleave it. So now this uh, foreign DNA is getting cleaved because of the restriction enzymes which are produced by the bacteria. So now this foreign DNA will not infect the bacteria, but the bacteria has to preserve its own DNA also. So what bacteria has done, that bacteria has methylated its DNA. So as you can see in this figure, that the own restriction sites are methylated. Now the EcoR1 is unable to cleave its own DNA, but it is able to cleave the this thing, bacteriophage DNA. So restriction methylase system. So this is the property of the restriction enzymes, that especially the type 2 restriction enzymes, mostly, that they cannot cleave the methylated DNA because the bacterial DNAs are methylated. That's why they protect themselves. Is it fine now?
Ma'am, I have one more question. So now
Yes.
when we say like when a person was a person was infected with COVID, so now COVID has different types of variations in it. So we have known that bacteria's outer membrane, it evolves itself towards the surrounding. So how come like it's related to restriction enzyme? Like
No. Yes.
after every like we have many vaccinations for COVID like Covishield and all, so how come Covaxin, so is it related with these enzymes, like do they act with the DNA and all, so how it is exactly?
So basically, you see, so viruses are very clever. And why viruses are so difficult to defeat is A, because most of the viruses have an RNA genome. Okay, so when you have an RNA genome, so if you, I don't know if you know or not, but when you have a DNA genome, so when DNA undergoes replication, so they have a very good proofreading activity. So if there are any mutations in the DNA genome, then because of the proofreading activity, then those mutations are repaired, or if it cannot be repaired, then that DNA replication is halted. But when we talk about RNA, then transcription or RNA does not have a very good proofreading activity. So because RNA does not have a very good proofreading activity, so whatever mutations that are occurring in the RNA, that will persist, it will not go. Now suppose that see, bacterial and mammalian systems are different, but I'm just telling you that suppose uh, there is one variant of the virus, coronavirus. Now we have made a vaccine against this variant 1. Okay, now this mutant this virus has evolved, and now it has made a mutation. And now because of this mutation, suppose we were treating covaxin. Now this covaxin was uh, directed towards this particular variant, but this covaxin will not be able to defeat this mutated virus, because this covaxin was made specifically for this version of virus. But here what happened was that luckily, we were very lucky that the mutations were happening, and that's why there were so many waves of corona, because of the different mutant strains that were coming. But eventually what happened is that the mutant strains that we had, they were of less and less this thing less and less harmful. That's why we were able to evolve. And also because uh, our immune system also got a little uh, adapted, and it also got a little more knowledge about the virus, and our immune system also evolved. But that is the problem, that the vaccine that you have, it can be specific for a particular strain of virus, or some few strains of viruses. But if the virus is mutating, and if there are some major mutations, then that vaccine is not useful. Do you understand? So
Yes, ma'am. Thank you.
so
Yes.
this restriction enzymes are specific to bacteria. We're talking about bacteria, but in general about the COVID, this is what has happened. Anyways, so the answer of
So
the
we can
question.
say that virus does not have their own restriction enzyme.
No, no, viruses does not have the restriction enzymes. Viruses are attacking. See, restrictions enzymes are present in bacteria. Okay, restriction enzymes, it is present in bacteria. Okay, so when I just give an example of bacteriophage. So bacteriophage are viruses that infects the bacteria. So basically, if this is a bacteria and this is its genome, any foreign DNA which is coming, so just like we have our immune system that attacks on the foreign particles that infects our cells, so bacteria's immune system is the restriction enzymes. So these restriction enzyme, it will cleave the DNA, but its own DNA would be protected because it is methylated. Okay, this is the bacteria story. Or the virus story is that viruses have DNA genome also, but mostly viruses have RNA genome. And if they have RNA genome, they are very prone to mutations, because RNA replication has not very good proofreading machinery, whereas DNA replication is has a very good proofreading machinery. So DNA is, when DNA is replicated, if there are any mutations, that gets repaired, or if they cannot repair, the DNA replication is halted. Therefore virus is able to mutate a lot, and that is a problem for us. So virus needs a host so that it can produce more of its viral particles. That is why virus, we say that virus is a link between living and non-living, because it is not completely living also, because it does not have its own machinery to synthesize its own particles. It needs a host. But it is not completely non-living also, because it does have a genome. So that's why it's a link between living and a non-living. Okay, so now we'll come to the next question. So in the restriction methylase system, RSM refers to what? Tell. What does RSM refers in restriction methylase system?
Ma'am, option A.
No. So the answer of this quite sorry. Uh, hmm. What should
Ma'am,
be
then
the answer
option
of?
B,
Yeah. So the answer
recognition,
is
then okay.
option B. So R stands for restriction, S stands for recognition, and M stands for methylation. So that means these are the three.
Ma'am,
Yes.
ma'am, so first it restricts, then it recognizes.
No, no. First, See, this is RSM does not define the sequence of the activity which happens in bacteria. So first it recognizes, then it see whether it has to methylate or not, then it restricts, then it cuts. So this RSM, in general we say it is RSM, but it doesn't mean that this is the sequence of the activity that a bacteria does. So first a bacteria recognizes the sequence, and then it says sees, sorry, first the restriction enzyme recognizes the sequence, and then it sees whether it is a methylated or a non-methylated one. So because it has to differentiate between self and non-self, right? So suppose if the DNA is hemimethylated, hemimethylated means one strand is methylated and one strand is not methylated, then it methylates the other strand. So that is the activity of M, that it checks whether the sequence is methylated or not. If the sequence is not methylated, then with the help of its restriction activity, it cuts. Okay. Now we come to the calculations. So the calculation for setting a restriction enzyme digestion. So before starting, just first tell me that what are the things that are needed for setting up a restriction enzyme digestion reaction? Anybody who remembers? Hmm? Anybody remembers what are the things that are needed for setting a restriction enzyme digestion reaction? What are the things that we need? Think logically. Digestion. We have to do digestion. So what we need first of all?
DNA.
Ma'am, DNA polymerase. Polymerase.
No, not polymerase. We have to digest our DNA. So we have to digest our DNA. So we need the DNA. Okay. Second thing is,
Restriction enzyme.
yeah, sorry, restriction enzymes. We need some restriction endonucleases. Then we need a buffer which is specific to the restriction endonuclease. And then to make up a reaction, we may need water also, sterile water or nuclease free water. And like sir has uh, told you in the class that
PSA.
you may need BSA also, but BSA is something which depends. Like BSA is not always needed, like as sir told, that it depends on the reaction to reaction. Like we in our lab, we never have used BSA because our reaction works in this way. So buffer should be 1x, BSA should be 1x. Okay, so these are the things that you need. You need BSA also. Chalo, BSA also we'll write, but buffer also 1x and BSA also 1x. Now, suppose from the genomic DNA, I wanted to clone a particular fragment, and this fragment I have isolated from the DNA, from the genomic DNA, and my reading was 250 nanograms per microliter. After isolation of genomic DNA, or DNA, that means after the isolation of our insert, this was the concentration. Now, how can I set the reaction? So, for setting a digestion reaction, so the DNA concentration is 250 nanograms per microliter. Okay, and we have studied that for digestion approximately 1 microgram of DNA is needed. So just tell me how much DNA we would need. The concentration of DNA is 250 nanograms and we need 1 microgram. So how much DNA has to be added in the digestion reaction mixture? Can anybody tell me? Anybody who wants to give a try? No? 1 microgram is how many nanograms? Anybody knows it? 1 microgram is how many nanograms? Hmm? Nobody knows? Basic calculation? 1 microgram is how many nanograms? Nobody knows here. Okay. So 1 microgram is 1000 nanograms. Fine. Now, the DNA concentration that we could observe after DNA isolation is 250 nanograms per microliter. What does it suggest? It suggests that in 1 microliter, we have DNA concentration of 250 nanograms. Okay. So, 250 nanograms in 1 microliter. So, I'm telling you in the very basic way. So, 1 nanogram would be in 1 upon 250 microliters. So 1 microgram, which is 1000 nanograms, would be 1 upon 250 into 1000. How much it will come? 4. 4. That means when we will use 4 microliter of DNA of this concentration, so 4 microliter of DNA will give us a concentration of 1000 nanograms or 1 microgram. Is it clear to everybody or not? Tell me, is it clear or
Yes,
not?
ma'am.
Yes, ma'am. Okay, so first thing for setting a digestion reaction is that we need the insert or the vector also. So anything minimum concentration should be 1 microgram, which is equals to 1000 nanograms. Okay, so whatever DNA concentration we have, accordingly we will calculate. So here we got our DNA as 4 microliter. That means 4 microliter in this case is equals to 1000 nanograms of DNA concentration. So first my DNA is 4 microliters. Okay, now I have to add buffers. Now which buffer I have to use depends on which restriction enzyme I am using. Okay, so I am using my restriction enzyme EcoR1, very basic restriction enzyme I am going to use. Now what buffer I need? So we have to check. So basically there are many kinds of buffers, like we have 10x buffer, we have R cut smart buffers. So these are basically the name of the buffers that are available. So I'll just show you. So we'll go to NEB cloning. Okay, this is a site. I'll go to NEB cloning. Now there is some problem. Let me see if it's some down or what. I don't know. I think it's showing some problem. If it won't work, anyways, we'll see. I think there is some problem. Just a minute. No, I think it has to be NEB cloning. Anyways, so you have to go to NEB cloning. I'll try again afterwards. So in NEB cloning site, you will type the name of the restriction enzyme and it will tell you that which buffer is the most suitable for that particular restriction enzyme. Okay, suppose my buffer comes at R cut smart buffer. So whatever buffers that are available, that are 10x buffer, but we have to use them as 1x. Fine. So what we will do is that our DNA is 4 microliter. Okay, and I want to set up a reaction of 50 microliter, suppose. Fine. So my buffer is R cut smart buffer, which comes as 10x, and I have to use it as 1x. So how much I will use? 5 microliter. So 1x of 50 is 5. Is it clear or not? You know how to calculate this? See, you know this formula? M1V1 is equals to M2V2. This is stock. This is working. So my M1 is 10. We have to calculate. M2 is 1 and the reaction is 50. So if I do, it comes 5. So that's why I have used 5 microliter. Is it clear or there is a problem? Tell me. 1x of a 10x buffer for a 50 microliters reaction is 5 microliters. Is it clear or not?
No, ma'am.
Somebody said no, ma'am. What is the problem? Tell me.
Ma'am, I cannot understand that procedure which you have done means on the calculation. Can you
Which part
please explain
you,
it again?
which part you couldn't understand? Tell me which part
Ma'am,
exactly.
ma'am, I mean that from the starting calculation.
See, for setting a digestion reaction, you need DNA. Okay, so here
Yes,
our case DNA is 4 microliters. This you could understand or not how we have calculated 4 microliters of DNA.
yes, ma'am.
Now we need buffer. Now, which buffer we have to use, it depends on the restriction enzyme. So, suppose I am using a restriction enzyme EcoR1. So, we have to choose a buffer which is compatible with EcoR1. So, we will go to, there is a site which is known as NEB cloning, and there are many other sites also. So, online we will check that which buffer is compatible with EcoR1. Okay. Now, we got to know that the name of the buffer which is compatible with EcoR1 is R-Cutsmart. This is the commercial name of the buffer. Now, buffers are supplied in the concentration of 10x. But whenever we are setting up a digestion reaction, the final concentration of buffers that we use is 1x. Okay. Now, how to calculate from 10x to 1x? The formula is M1V1 is equals to M2V2, where this represents the stock and this represents the working. So, the stock concentration is 10. So, we will use 10. V1 we have to calculate. The working M2 we want is 1. And for how much reaction? We want the overall reaction of the overall volume of the digestion reaction as 50 microliters, suppose. So it is 50. Now you calculate V is equals to 5. That means that when you will use 5 microliters, it will give a 1x concentration for a 50 microliters reaction. Could you understand it now or not?
Ma'am, I have one doubt means why do we use ma'am like 10x to 1x in here?
See, so the buffers that you get, they are very concentrated. So you cannot use that concentrated buffer. You have to dilute it. Basically, the meaning of this is that you are diluting your buffer. So buffer is means the concentration of the buffers is 10x. So that means whenever you are doing some biotechnology experiments, you have to always remember that biotechnology solutions mostly they are stock and you have to prepare a working solution. So not always you will get a working solution. So from a stock you have to prepare a working of whatever dilution you need. So for a digestion reaction we need a 1x concentration. So for 1x concentration you have to uh, calculate how much is needed, but it is very easy. If you have a 30 microliters reaction, so 1x would be 3. 40 microliters, 1x would be 4. 10 microliters, 1x would be 1. So I just showed you the calculation to explain the logic behind it. But whatever is the total volume of your reaction mixture for digestion, 1x would be, if total volume is 30, 1x would be 3. Total volume is 50, 1x would be 5. Is it clear?
Yes, ma'am.
So now we have DNA, we have buffer. Now we need the restriction enzymes. So suppose restriction enzyme we add. How much restriction enzyme we have to add? What has sir given you? So basically, mostly we add restriction enzyme 1 microliters, and then 50 microliters reaction we have to set, and then we will add NFW. So matlab water. So how much water we will add? See. This is 4 plus 1, 5. Total DNA is 4. Buffer is 5. Restriction enzyme is 1 microliter. And the total reaction mixture has to be 50 microliters. So NFW, how much NFW you have to add then? You have to calculate. This is 5, 5, 5, 10. That means 40 microliters of NFW you have to add. Is it clear or not?
Yes, ma'am.
So what?
Yes,
So suppose if you have to set up a 50 microliters reaction, then you will calculate all the reagents that you have to add, and whatever is the remaining part, that much NFW you have to add. Okay, but when you are setting up a reaction, always you have to add NFW first, then you have to add the buffer, then you can add the DNA, and then you can add the restriction enzyme. Always remember, whenever you are doing any kind of experiment where enzymes are involved, it can be restriction enzyme, it can be DNA polymerase, it can be any kind of tag DNA polymerase. Enzymes are always added at the last. Why? Because we don't want to inhibit or uh, decrease the efficiency of enzymes at any cost. So what is the logic is that you add the enzyme, immediately you put it for the reaction, immediately you put it in the PCR machine. So enzymes are always added in the last. So this is the way how you set up a digestion reaction. So is it clear to you how to set a digestion reaction? Has anybody any confusion? Because this is very not complicated, but you won't get any other exposure for setting a digestion reaction, because it depends on the experience, if you have any experience of setting up. So if you have any doubt, you can ask. Otherwise it's difficult for you to
Ma'am,
get in books or anywhere else. Yes.
ma'am, can you explain once more the calculation of DNA?
Okay, this is the last time I'm explaining DNA calculation. No, DNA means the that how 4 microliter came, that you wanted to know.
Yes, ma'am.
Okay, so see, last time everybody listen carefully. So for setting a digestion reaction, we need DNA. Okay, first thing is we need the DNA insert. Now DNA would have some concentration. So suppose the concentration of DNA is 250 nanograms per microliter, which means in 1 microliter 250 nanograms of DNA are present. Now I want 1 microgram of DNA for my digestion reaction. 1 microgram is equals to 1000 nanograms. So in 1 microliter 250 nanograms of DNA is present. This is the meaning of this particular thing. So 250 nanograms in 1 microliter. So 1 nanograms in how many microliter? 1 upon 250. So 1000 nanograms in how many microliter? 4. That means when we will take 4 microliter from this concentration of DNA, it will give me 1 microgram. Is it clear now?
Yes, ma'am.
Okay. So we have DNA, which is equals to 4 microliter. Then we need buffer. So buffer depends on the enzyme that we are using. If I am using EcoR1 from the net, I could find out that for EcoR1, the commercial buffer that is used is R-Cut Smart Buffer. Then it is the commercial name of the enzyme buffer. All the buffers are supplied in 10x concentration. So we have to dilute it. So from 10x, we need the final concentration should be 1x. So we will calculate for a 50 microliters reaction, 1x would be 5 microliter. Then we will add the restriction enzyme. Restriction enzyme, we will add 1 microliter. Then we will see 4 plus 5, 9 plus 1, 10. So how much NFW you have to add? You have to add 40 microliters, because overall the reaction is of 50 microliters. So like this is the calculation for restriction digestion. Okay, now if you have still you have doubts, you can again go through it whenever I'll upload it in the YouTube video. Now I'll go to the next question, because already we have given a lot of time to digestion. Fine. Star activity is shown by enzymes when? What should be the answer? First of all, tell me what is star activity. What do you understand by star activity? Anybody knows what is star activity? Tell me. Do you know what is star activity? Nobody knows. So, have you really read uh, the lectures of this week or not? Because it doesn't look like you have read anything this week. Because this is very basic. You don't know what is star activity. Sir has already taught in the class what is star activity. Hmm? Nobody knows here what is star activity. So
Ma'am,
basically,
the restriction enzyme,
hmm, restriction enzymes what?
which are cleaved and are not identical.
No, 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 no. You're talking about isosisomers, neosisomers, isocodomers. No, that we're not talking about here. So see that suppose restriction enzyme EcoR1, just given an arbitrary example. So EcoR1 has a specificity of GAATTC. Now, star activity means star activity is an activity which is shown by restriction enzyme. Sometimes what happens because of certain conditions, the this EcoR1 will not only recognize GAATTC, it can also recognize GAATTA or GAATTT or GAATTG. That means the non-specific activity which is shown by restriction enzymes is known as star activity. That means apart from recognizing its own sequence, it will also recognize similar sequences. That is known as star activity. Now, star activity is shown by enzymes when sometimes what happens that the glycerol concentration in the reaction mixture is more than 5%, yes, and if the enzymes added is more as compared to the DNA, yes. So the answer is A and B. So what is glycerol? So basically you have restriction enzymes. I don't know how many of you have seen restriction enzymes. So if this is a vial of restriction enzymes, so these restriction enzymes are suspended in glycerol so that they do not freeze. So that means that you should always add an optimum concentration of restriction enzymes to your reaction mixture. If you are adding restriction enzymes more, then it is a problem. Then your enzyme can show star activity. And if the glycerol concentration is more than 5%, then also your enzyme can show star activity. So star activity is basically the non-specificity of enzymes. Is it clear to everybody? Okay, fine. We'll move to the next question. Which enzyme prevents the recircularization of DNA and how? Tell me the answer. Which enzymes prevents the recircularization of DNA? What is recircularization of DNA at first place? Tell. Hmm? Are you guys really reading something or you're not reading at all? It is looking that this week you haven't studied anything because you're not able to answer any questions. Option B. What is option B? Which enzyme is present? No, no, no. This is not the correct option. B is not the correct answer. Try. Try more. Which enzymes prevents the recircularization of DNA? So the answer is C.
Ma'am, ligation.
No, no. Answer is C, alkaline phosphatase. So, what is recircularization? So, basically, that suppose this is my vector, okay. Now, I cut with it restriction enzyme. Now, we know that if this is a linear DNA, at the 5 prime end, you have a phosphate group, and at the 3 prime end, you have an OH group. Okay, so what happens that when you cut this restriction enzyme, so here this is there is suppose phosphate, and here there is OH. Okay, now there is a possibility, then again it will get stick and again it will form a circular, again it will religate. So recircularization means the religation of the vector molecule. Okay, so there is a possibility that this vector can religate because there is one P and there is one OH. So what we can do is, what people do is that they treat their vector with alkaline phosphatase. So alkaline phosphatase remove the 5 prime phosphate group. Now once this 5 prime phosphate group is removed, then there is nothing here. So then this vector is not able to religate or recircularize itself. So phosphatase means phosphatase, matlab it will remove phosphate. Phosphatase means it is an enzyme that removes the phosphate. From the name itself, it is clear it will remove the phosphate. So it removes the 5 prime phosphate group so that the vector is not able to religate um itself. So that is the recircularization of DNA. Is it clear?
Yes, ma'am.
Okay. Now we'll go to the next question. So this is the calculation for setting a. Acha, this should be ligation. So we have already discussed digestion. We will discuss ligation, but ligation we will discuss at the end. First, we'll do the questions. So now in lecture 2, we have studied about the different kinds of vectors. What is the criteria of a good vector? What are the components of vector? What are the different types of vectors? What are bacterial plasmids? In bacterial plasmids, we have studied about PBR 322, PUC 19, etc. So one thing you have to remember is that vectors, basically, what is the function of vectors? So whenever we are dealing about cloning, this module is about genetic engineering. So genetic engineering means that you are inserting a foreign DNA into any other organism or in any another host. So for inserting a foreign DNA, you have to first cut the DNA and you need a vector. So you need an insert. I'll just show it with the help of the diagram that sir has made. Yeah. So for producing, for genetic engineering means that you are producing a genetically modified organism. So for producing a genetically modified organism, what you have to do, you need the genome. So from this genome, you will isolate the gene of interest. Okay. Now here only I was talking about that, suppose the concentration of DNA is 250 nanograms or whatever. Then you will cut the DNA. And again you have a plasmid, you will digest it and you will cut the plasmid. Now the DNA and the plasmid you will cut with the same restriction enzyme. That means we have already dealt a question like this in our previous lectures. Suppose if you have cut your DNA with eco R1, then you will cut your plasmid also with eco R1. Okay, so first step is amplification of the gene of interest from the whole genome. You want a particular piece of fragment, so you have amplified that particular piece of fragment only, and you have your commercial plasmid. You have digested your gene of interest and you have digested your plasmid, and then you will ligate it, and then you will transform it, and then you will screen it. Okay, so we have discussed about digestion and how to set up a digestion reaction. Now here, before going to how to set up a ligation reaction, we have to see that what are the different types of plasmids that we can use for producing a genetically modified organisms. Is it clear? So the different kinds of plasmids that we can use, plasmids are basically the vectors. So these vectors should have the ability to carry the foreign DNA, and it should have the ability to replicate. So replicate means that it is able to make more copies of itself. So when I say that a vector is of high copy number, okay, if I say that a vector is of high copy number, it means that it is producing more copies inside the host. Okay, it is producing more copies. And if I say that a vector is low copy number, that means it is producing less copies. So that means a high copy number vector means it is replicating more. A low copy vector means it is replicating less. Okay. So this is the two major properties of a vector. So now what are the criteria of a good vector? How a good vector should be? It should be of low molecular weight in size. It should have phenotypic changes, means you have some phenotypic changes so that it can be screened. It should have unique restriction sites. It should have multiple cloning sites. It should have a high copy number. So these are the criteria of a good vector. Now, accordingly, we'll see the question. The following describes the criteria of a good vector. Pick the odd one out. Tricky one. Now, tell me the answer. These are the some options. Tell me the right answer.
Ma'am, option A.
No, option A is not the correct one. No. Just a minute. Someone joined. Yeah.
Ma'am, option B.
Okay, so we have Mamita. So Ashwarya is saying option D, and Nandini is saying option B. Let's see. Option D is high molecular weight. No, option D is wrong. Option B is correct. So Ashwarya has given, no, Nandini has given the right answer. And Mamita, you have raised your hand. Yes, what is the query? You have raised your hand, Mamita. Anyways. So yeah. So here you have to see that you have to choose an option which is, see, odd one out means, odd one out does not necessarily means that it is the wrong option that you have to choose. This is how your MCQs trick you sometimes. So you see the options high molecular weight and low copy number. No, it should be reverse. It should have a high copy number and it should have a low molecular weight. So this is not, this is incorrect one. This is an incorrect one. Low molecular weight and low copy number. Low molecular weight is okay, but you should have a high copy number. So this is also an incorrect one. Multiple cloning size and phenotypic changes. Yes, it should have. It should give some phenotypic change so that it can be screened, and it should have multiple cloning sites. So this looks like a right one. Low copy number and multiple cloning sites. Multiple cloning sites is okay, but not low copy number. It should have a high copy number. That means in this question they have actually given you more of wrong, and the odd one is actually the right. So basically this odd one out is nothing. This is just to trick you. You just have to pick the correct one. Here the correct one, obviously in MCQs the correct one is the odd one because all others are wrong. This is just to trick you. Anyways, that's why you should know the concept. So a vector, it should have a high copy number. That means it is able to replicate more. It should have a low molecular weight. It should give some phenotypic changes. It should have some phenotypic changes. And it should have multiple cloning site, MCS. So the answer here is B. Now, question number 6. Sudha was trying to express a DNA fragment in yeast. Okay. She used PBR322 as the vector. However, she was unable to express the gene. Identify the most suitable explanation. A. The DNA fragment was not properly ligated with the vector. The DNA fragment was cut with type 1 restriction enzyme. She has used a prokaryotic vector to express the eukaryotic gene. None of the above. So what is the answer? Think and answer.
Ma'am, she has used a prokaryotic vector to express a eukaryotic gene.
Yes, correct. So, sorry. Yeah. Huh. So she has to clone a DNA fragment in yeast. Okay. That means, but she has used a PBR322 is a bacterial plasmid, and she has to clone it in yeast. So basically, the gene is from yeast, from a eukaryote, and she's trying to clone it, express in a prokaryotic vector. So is it possible? No, because for a eukaryotic gene you need a eukaryotic vector. Why you need a eukaryotic vector for expression? Because eukaryotic vector will only have the components that are needed for the expression of a eukaryotic gene. Prokaryotic vector will not have. So the answer is that she has used a prokaryotic vector to express a eukaryotic gene. Now, question number 16 is the selection markers present in PBR322 and PUC19 are what? Tell me the answer. Think and answer. What should be the answer of this question? Anybody? So there are some. Okay. So B and A are the two options. Mamita, you are raising your hand, but you're not saying anything. B and A. B is both have. No, B is not the correct answer. A is the correct answer. Okay. So the correct answer is A. Now you have to see that the first vector. Just a minute. Um, so who has given the correct answer? Nandini. So can you explain the reason? Can you explain the reason why you said? Tell me. I want the class to be interactive, so please respond. So Nandini, you have given the right answer. So can you tell the reason why you chose the answer, option number A? Hmm? No one is responding today. I think everybody is in the mood of holiday today. Nobody wants to study much. Hmm?
Ma'am, can I try?
Ha, anybody can try.
Ma'am, so in PBR 322, there is a selection marker known as antibiotic selection.
Yes.
But in PUC 19, there was one theory of white and blue-white colonies uh,
Hmm.
in screening.
Yes.
So, so this may be the reason for option A.
Hmm. Yes, you are correct. So basically, the first cloning vector which is made is PBR322, and then after PBR322, there is another vector which is PUC19. So basically, PUC19 have a selection criteria which is known as blue-white screening. Fine. So blue-white screening. So basically, you have to screen the bacteria that has the colonies. Okay, I'll just explain. I think. See, so you have, this is the DNA and this is the vector. You have ligated it. Okay. Now you introduce this vector in a host. So my ligated vector. So I will introduce my ligated vector in a host. Okay. So suppose my host is, let's say, a bacteria. Just let's say. Now I want to select only those bacterial colonies that has the vector. Okay. So there I have to screen only those bacterial colonies that has the vector. Now for that, if I have a plate here, I will have we have many different criterias for screening. The first one is the antibiotic resistant. That means antibiotic selection markers. So what happens that in your bacteria, if your bacteria has an ampicillin selection marker, that means this bacteria is resistant to ampicillin antibiotic. So when this particular vector, when this colony will grow, this is a see, I think I have to explain from the very basics. So, after ligation, you have a vector plus DNA kind of a thing. You have ligated. But there are some chances that the ligation may not work properly. So, that is different that we leave. So, now after ligation, you will introduce this to a host. Okay. Suppose my host is a bacteria. Now this bacteria, you will keep this bacteria to grow. So in my plate, there would be many colonies. Okay. And this plate is a agar plate which has ampicillin in it, suppose. So ampicillin will not allow this bacteria to grow because bacteria is a kind of infection, na. So ampicillin is a gene which will inhibit the growth of bacteria. So how then this bacteria will grow? Only those bacteria will grow that have a ampicillin resistant gene. That means those bacteria that are resistant to this ampicillin, and those bacteria are the cloned one, because in our this thing vector we have an antibiotic resistant gene, that is ampicillin. So the only those bacteria will grow which will have the resistant gene of ampicillin. Is it clear to you or not? Is this particular thing
Yes,
clear to
ma'am.
you? Okay, just a minute. Hmm. So this is your antibiotic selection criteria. So antibiotic selection criteria means that the plate in which you are plating your colonies, host cell colonies, your host cell will only grow if your host cell has a plasmid, because that plasmid has your ampicillin, your antibiotic resistant gene, because bacteria is a kind of infection and this plate already has ampicillin in it. This plate already has ampicillin, so it will inhibit the growth of bacteria. But if the bacteria has the vector which has the resistant to this ampicillin, then the bacteria will grow. So PBR 322 has the antibiotic selection marker, whereas PUC19 has a blue white screening method. Blue white screening we are not going to discuss. We'll see if we have time, because it is going to take a lot of time. Okay, so next thing is lecture 3. In lecture 3 we have studied about isolation of plasmids, some eukaryotic vectors, what some yeast vectors, baculovirus plasmids, mammalian vectors, and bacteriophage lambda based vectors. So basically eukaryotic vectors can be your yeast system, yeast vectors. It can be your baculovirus, that means that infects the insects. You can have mammalian system and bacteriophage. So bacteriophage we have already discussed. Bacteriophage is a virus that infects the bacteria. Now we'll see this question. The composition of resuspension buffer for the isolation of plasmid is, what is the answer? What is the answer? So the answer is option A, that is 50 millimolar glucose, 25 millimolar trisethiol, and 10 millimolar DDT of pH 8. You should go. There is nothing to explain because Nandini, because blue white screening in PAC 19 and antibiotic selection. Fine. Now we come to the ninth question. What are the three bands observed after the isolation of plasmid? Identify in the order of elution. Anybody know the answer of this question? I think this week you haven't studied properly because you're not very active in the class, answering
Ma'am,
very few
uh,
questions.
covalently, uh, covalently closed circle, open circle, and supercoil.
So you are saying option number B. This option you're saying, right? So let us just find out. We'll see in the lecture.
Ma'am, it's option B.
Hmm. So isolation of plasmids. So that what does it mean is that whenever you are isolating a plasmid, okay, so whenever you are isolating a plasmid, so plasmid, and when you will run it across gel, it will give you three bands. So first one, in the order of elution, the first one that would be eluted is supercoiled, then it would be open circle, and then it would be covalently closed circles. So because if it is super coiled, then it is able to move faster. When it is a linear or nicked, it would have a lesser speed. When it is a covalently closed circle, then it would migrate with the least speed. So first elution would be super coiled, second would be open circle, and third would be covalently closed. Is it fine to everybody?
Yes, ma'am.
So now here it is actually, see, you are saying first is covalently closed circle, but first is actually supercoiled. So the answer should be first is supercoiled, second is open circle, and third is covalently closed. So you just have to understand the language. I think you know the concept. Just you have to know the language. Order of elution means what is the first thing that is getting eluted. So first thing that is getting eluted is the supercoiled, and then your open circle, and then your covalently closed. Okay, now we'll go to the next question. What kind of vectors are used for stable gene expression? Plasmid, PUC19, integration plasmid, episomal vectors. Hmm.
Plasmid vectors.
No. So one thing that you have to understand is that there are two kinds of gene expression. Okay. So the two kinds of gene expressions are, the two kinds of gene expression are transient gene expression, and the second one is stable gene expression. So what is the meaning of transient and stable? So suppose if this is your host and this is the host DNA, okay, in transient expression what happens, that external DNA, it exists as an episome. That means it is not integrated with the host genome. Whereas in stable gene expression, what happens, that whatever DNA is you are getting setting, it will integrate with the host genome. So suppose if red color is your host genome, so your DNA, it is going to get integrated with the host genome. Whereas here the DNA would be present as an episode, matlab it is not integrating. So here it will be present for only this generation, only one generation, this generation. So whenever it will replicate, your this DNA is gone. But in stable, for whenever it will replicate, it is present for in subsequent generations also, your insert is present. So it is a stable because in subsequent generation also your insert is present. Whereas in transient it is not stable because the DNA is present only in that particular generation. After replication, the external DNA is gone. Is it clear, or anybody has any problem? Tell me. Hmm? Is it
Ma'am,
clear?
so why can't it be plasmid?
Why can't?
Why can't it be plasmid in this case?
Plasmid, see, anything. See, plasmid is just a vector, is just a carrying molecule which is carrying your DNA. So this is plasmid is a genetic material only. This is actually a plasmid only. Matlab this is a genetic material only which is getting integrated. It has to integrate. If it is able to integrate with the host genome, it will produce a stable gene, stable expression. That means the gene of interest is expressed in subsequent generations also. But in transient expression, the gene of interest is not expressed in the subsequent generations. So for stable gene expression, we need which kinds of vectors? Which kinds of vectors we need for stable gene expression?
Which can be integrated into the.
Yes, so we need integration vectors. For stable gene expression, we need integration vectors. So these are not integration vectors. So yeast vectors, if you have studied, then yeast vectors exist in two forms. They are episomal vectors also and yeast vectors also. You see yeast vectors. So yeast vectors, there are three types of yeast vector system. The first one is the episomal vector. So basically episomal vector will not integrate with the host genome. It will be present extra. Whereas integrating vectors. So what is there? Episomal yeast vectors are present as extra chromosomal DNA and they are unstable. This can be overcome by integration of vector in the host genome. So that means integration vector, they will integrate with the host genome. Whereas episome is basically the episomal DNA is the extracircular DNA. Matlab DNA apart from the genomic DNA, the extracircular DNA which is present in a cell, that is your episomal DNA. So integration vectors will provide us stable expression. Okay?
Okay, ma'am.
Fine. Now shuttle system of vectors is useful because it helps to digest the gene from both prokaryotic and eukaryotic, helps in screening, helps to propagate the vector in both prokaryotic, eukaryotic environment and all of the above. So what is the answer? Hmm? What is the meaning of shuttle? Tell me. So basically, shuttle. What is the meaning of shuttle? Fine. So shuttle means in normal to and fro. Matlab in both the places it is going. So you have, I don't know how many, you have buses, you have shuttle buses who takes you from one place and you drop it there and then again from here it takes you here. So like this, in both the places it is going. So shuttle vectors are the vectors basically that are able to propagate in prokaryotic environment also, and they are able to propagate in eukaryotic environment also. So that means for propagation, what is needed for propagation in a vector, how a vector can propagate matlab how a vector can replicate when it has the origin of replication. Only then a vector is able to replicate na. So if you have now these origin of replications, they are specific for prokaryotes and they are specific for eukaryotes. That means for prokaryotic system you have a different origin of replication, whereas for eukaryotic systems you have a different origin of replications. So a shuttle system is a shuttle vector is designed in such a way that it has origin of replications for prokaryotes also and origin of replications for eukaryotes also. So that means that whether you transform this particular vector in a eukaryotic host cell or in a prokaryotic host cell, in both the systems it can work. So shuttle system of vector is useful because it helps to propagate the vector in both prokaryotic and eukaryotic environment. Is it clear? Or anybody
Yes,
has
ma'am.
any doubt, you can ask. If anybody has any doubt, you can ask me. Okay, so shall I move to the next question? I'll move to the next question now. The biosynthetic pathways used as a selection markers in yeast are, so basically in yeast, till now we have studied that selection markers can be antibiotic genes. Okay, you can have blue white screening. These things we have studied. This is present in PBR. This is present in PUC19. Okay, and another kind of selection criteria is we can use the biosynthetic pathways. Okay, so the two in yeast basically for the selection criterias, uniquely there are biosynthetic pathways. And the two biosynthetic pathways that are used in yeast are D, that is tryptophan biosynthetic pathway and uracil biosynthetic pathway. Okay, next question is dash is required for transmission of virus from one host to another in the case of baculovirus. What is the answer?
Polyhydrin.
Yes. So the answer is, correct answer is polyhydrin. Now, next question is the circularization of bacteriophage is caused by digestion with restriction enzyme that produces sticky ends, cause sites present in the middle of the genome, binding of the adapter molecules at the end, and none of the above. So what is the answer?
Ma'am, C.
No, C is not the answer.
Ma'am, E.
No. So the answer is none of the above. Why? So basically, bacteriophage is present in whenever it is in the linear form, it has sticky ends at the end. At the end of the bacteriophage, you have six sticky ends, and these sticky ends are known as cohesive sites. Okay, and in short, these are known as cos sites. So because of these cos sites, the bacteriophage, it kinds of make itself circular. But here I have given cos sites, but I have written in the middle of the genome. But cos sites are present at the end, because if they are at the middle, then how will it be able to join? So it can, this DNA, it can join only when something sticky is present at the ends, no? If something is present in the middle, then how it is able to join? So the sticky thing that is present at the end that causes this to form a circular molecule are the cohesive sites, or in short, we call it as cos sites. Okay? So the answer is none of the above. Now, next question is selection criteria of bacteriophage lambda based vector is. So basically the unique selection criteria for lambda based vector, because now in general if you see that most of the vectors they have some antibiotic resistant genes. That's why I have written antibiotic resistant gene only. So this is not the answer. So what is the answer here? For bacteriophage lambda based vector, what is the selection criteria? Tell me. Selection criteria of a bacteriophage lambda based vector is. So till now, how many selection criteria we have studied? Antibiotic resistant screen, blue white screening we have also studied. We have also studied that some vectors uses the selection criteria of biosynthetic pathways. So bacteriophage uses the criteria of plaque formation. So viruses, bacteriophage are virus. Viruses, they produces plaque. So the answer is plaque formation. Okay. So
Ma'am,
yes.
ma'am, in the previous question, ma'am, why option A is not correct?
Yeah, see, it is not correct because the peculiar property or the most distinctive property of bacteriophage is that it has cos sites in the ends which causes the circularization. So basically digestion with restriction enzymes that produces sticky ends, it will obviously cause the it will cause the circularization, but it is not needed in bacteriophage because they already have inbuilt sticky ends. Could you understand? You digest with restriction enzymes. Why? Because you want some sticky ends. But when you already have sticky ends, then there is no need of digestion. So bacteriophage already has cos sites. So there is no need of digesting it with an enzyme so that you can have a sticky ends. Is it clear?
Yes, ma'am.
Okay. Now next question is ions required for the activity of type 2 restriction enzymes are. What kinds of ions are required for type 2 restriction enzymes? Think. So the answer is D. Mg2 plus. You have to read it. Single stranded unpaired extensions formed by restriction enzymes upon cleavage is called as what? Tell me. Single stranded unpaired extensions. What are these?
Ma'am, blunt ends.
No. Blunt ends can't be. Why? Because they are sticky and they are single stranded. Suppose this is your DNA, and if you are producing blunt ends, so blunt ends are always double stranded. Whereas when you are producing sticky ends, if you are digesting and you are producing sticky ends, so sticky ends are there is a portion which is single stranded, this portion is single stranded, this portion is single stranded. So here that is why it is written single stranded unpaired. So this is the answer is sticky ends. Next question is blue white screening in baculovirus can be facilitated by ampicillin gene,
Laxi gene.
LAGZ gene. So for blue white screening, which is what is needed, LAGZ gene is needed. Dash determined the sequencing of lambda phage DNA. I think it was not given. The answer is Sanger. I don't think it was given, but the answer is Sanger. So Sanger determined the sequence of lambda fast DNA. So you know the principle of blue-white screening or not? How blue-white screening works? So I will suggest you to just first read about blue-white screening. Okay, you read about blue-white screening because we will discuss it in the next class, but I will only discuss it when you have a basic knowledge. Otherwise, it would be very difficult for me to uh, make you understand the concept. At least you should have a basic idea about what blue white screening is, and then we can discuss it in the subsequent class. Also, I want to tell you one thing that I could see your performance is not up to the mark in this class. So it's fine that you don't know the answers, but your concepts or at least the basics should be clear. So I don't know how much you have studied this week, but you have to really gear up if you want to clear the certification course or just leave about the certification course. This is a kind of these are the kind of classes that you won't get in your normal college routine, okay, where you have so much of doubt clearing sessions and you have so much of thoroughly made uh, course structure by sir. So if you have enrolled in any course like this, you should try to take the full advantage of it. You should try to get the 100% the maximum out of it, because that is a waste. It is of no use that you are sitting here for 2 hours, that too on a holiday, and you don't have the basics, you haven't read about the things in your last week. Then it would not make any difference in your knowledge. So I can explain you a lot many things, but my energy and my enthusiasm will depend upon how much you are responding. So when I see that you have read, you have at least some basic idea, then I would also be more enthusiastic to explain you a lot of other things as well. So it all depends on how much you would reciprocate. Accordingly, I will do my job. Fine. So, anyways, I will always come on the Tuesday and I will always discuss the questions and the topics that has been covered in the last previous week. But if you want to get the most out of it, you have to read. So you should. I just hope that in the next online class, you people will at least read twice. At least you should go to the PPTs twice, or at least before the class you should have your ppts open in your laptops, so that you can just revise it before coming to the class, because it is helpful for you only. It is not going to benefit me in any way. Anyways, we'll just move to the subsequent question. Dash restriction enzyme cuts the only methylated DNA. Which restriction enzyme cuts? So till now we have discussed that restriction enzymes where a DNA is methylated, then it will not cut. But there are certain class of restriction enzymes that cuts only the methylated DNA and not the unmethylated DNA. So such enzymes are also present. So what are those enzymes? Which enzymes cuts only methylated DNA? Hmm? Tell me. Think. Try to think.
Ma'am, option A.
No. The option is type 2M. See, even if you don't have any idea, you can get a basic idea from the fact that something like M is written. So maybe, so basically type 2M are the enzymes that cuts only the methylated DNA. Okay. Now after replication, mutated DNA is repaired by which mode of replication? So DNA has what mode of replication? DNA replicates by
Semi-conservative mode of replication.
Semi-conservative.
yes. So the answer is, so even for the repair, it will use the mechanism by which it replicates. So it replicates by semi-conservative mode of replication. Now, next question is a typical restriction enzyme naming is based on. So, suppose if it is written ECO-R1. So, what does E stands for? What does E describes? E describes the
Option A, ma'am.
yes. Option A is the correct one. So, E describes the genus. Okay. CO describes the species. R describes the strain. And 1 describes the order of identification. It was the first restriction enzyme. So, EcoR1. Then the T for DNA ligase enzyme was isolated from. So, this enzyme is isolated from virus. Next question is, sorry. Cloning of blunt end is facilitated by. There can be more than one correct answers in this.
Option D, ma'am.
Option D. No, no. It can either be adapter molecules or linker molecules. So I'll just explain you, and then we have to do that particular thing also. So anybody remembers that where this adapter and linker concept? Here only it is given. Adapter molecules and linker molecules. So what are linker molecules and what are adapter molecules? Anybody has any idea after seeing the PPT?
They ligate at the cohesive end of the strand.
No. Okay, I'll tell you. So, I'll just explain you in the PPT itself. Correct. So, we know that restriction in basically type 2 restriction enzymes basically produce sticky ends, but two kinds of ends can be produced after the digestion with a restriction enzyme. The first is the sticky ends, and the second one are the blunt ends. Okay, so sticky end cloning is easier than blunt end cloning, because if this is your sticky end of your DNA and this is the sticky end of your vector, so this can, if this is your 5 prime, sorry, it is your 3 prime and it is your 5 prime, and this is your 5 prime, it is your 3 prime, it will form the complementary and it is able to ligate. So sticky end cloning is easy, but if you have a blunt end like this, then to clone a blunt end is difficult. So there are certain ways by which we can modify the blunt ends so that they become sticky ends. Okay, there are certain ways. So the two ways are ligation of adapter molecules and ligation of linker molecules. Okay, so now you can see that if these are the adapter molecules, this is the alkaline phosphate, sorry. So what we can do is that if this is your blunt end, you add a particular extra some nucleotides. Now this nucleotide has the site for a restriction enzyme which will produce sticky ends. See EcoR1. EcoR1 produce sticky end. So when you digest this particular added DNA with EcoR1, it will produce sticky ends. So that means that you have blunt ended molecule. You have added an extra insert at the end of this DNA, and these inserts have restriction sites for a sticky end producing restriction enzyme. So this will produce sticky ends now. Is it clear? Are linker molecules clear to you or not? Tell me. I think there is some question. Mamita, you are raising hand but you're not saying anything. Anyways, so what is the problem, Mamita? You are raising hands quite often but you're not telling anything. Tell me. Is it clear or not? So basically, linker and adapters are the ways of transforming a blunt ended DNA molecule to a sticky ended DNA molecule. So suppose this is your DNA and this is a blunt end. Okay, you are adding certain bases at the end, and now when you will digest this particular short stretch of nucleotides, it will produce a sticky ends. So now at the end you have sticky ends in your overall molecule. Okay, but there are possibilities that suppose if you are digesting it with eco R1, so eco R1 sites can also be present in between this DNA. There can be a possibility. So when you are digesting it with eco R1, you are not only producing a sticky end here at the end, but you're also fragmenting your own DNA. So it may cause problem. So the next strategy that is used is that you ligate certain molecules that already have the cohesive ends, that already have the sticky ends. That means there is no external digestion that is needed. They are already digested and they already have sticky ends. Okay, so those molecules that already have sticky ends, those are known as adapter molecules. Whereas linker molecules, so linkers you have to first add and then you have to digest it. Whereas your adapter molecules, they are already digested and you just have to attach. See, this is your DNA. Okay, and these are your adapter molecules which already have sticky ends, and this is a blunt ended DNA molecule. So when you add this, then you have a vector which has sticky ends. Whereas in linker molecules, what you have to do, see, these are the this is your foreign DNA and this is your linker molecule that you have added. These are the two linker molecules. On both the sides you have added, these are your linker molecules. When you add and then you treat with eco R1, so it will produce sticky ends. But the problem with linker molecules is that suppose the site for eco R1 is also present somewhere in the middle of the insert, then your insert can also get fragmented. Therefore you can use adapter molecules which are already sticky ended. You just have to ligate it and that's it. Is it clear to you or not? Hmm?
Yes, ma'am.
Okay, just a minute. Okay. Okay. So now we will do the calculation for setting a restriction enzyme. No, calculating for setting a ligation. We will see how to set up a ligation reaction. Okay. So now you have a digested insert. You also have a digested vector. You have to ligate it, and then this ligate would be transformed to a host cell, and then it will form colonies, and then these colonies you will screen. Okay, so this is the basic general protocol. Now for setting up a ligation reaction, mm, just a minute. Hmm. What you need is, you need vector, you need insert, you need your ligase buffer. So always whenever you are setting up any reaction, you always need buffer. For digestion, you need digestion buffer. For ligation, you need ligation buffer. You need your enzyme, suppose T4 DNA ligase, and you need nuclease free water. Okay, so these are the things that you need. So you should always try to have the vector to insert concentration as maybe 1 is to 3 or 1 is to 6 can also work. That means that for one vector there should be 3 molecules of insert, 3 insert. So 1 is to 3 means, sir has also explained you that the ratio of vector is to insert should be 1 is to 3. That means insert has to be present in more amount than the vector. Is it clear? This is the ratio in which you have to take the insert and the vector. For every one vector there should be 3 insert molecules. Like this you have to take. Then you have to take the nuclease free water. First you will add the nuclease free water, then you will add the ligase buffer, then you will add your vector or insert, then you will add your insert, and in the end you will add your enzyme. So for a ligation reaction, these things are needed. Again, mostly the buffers that are provided, especially for cloning, mostly they are 10x only. So depending upon the reaction, you can use, suppose if you have to use 1x, then accordingly you can calculate. Is it clear? Okay, so now for the last 15 to 20 minutes, I will take the questions from your end like we do in all other classes. So now you tell me that what part you could not understand in the last week. If you have really read something, I don't know. Tell any topic, any part that you could not understand in the last this thing class, the last week's classes, lectures, lecture 1, 2, 3. Hmm? So nobody is saying anything. That means either you have learned everything or you have read nothing at all. Any topic that you want me to explain again from the lectures? I'll just go through the lectures. Enzymes in molecular cloning, restriction methylase system, setting up restriction reactions we have already studied, ligase, phosphatase, linker and adapters also we have discussed. Then we have discussed the criteria of a good vector, components, the different vectors. Then we have studied about the different kinds of plasmids, the isolation of plasmids, the bands, the eukaryotic vectors. The eukaryotic vectors can be of yeast based, phage based, and mammalian based vectors. Yeast based vectors can be episomal or integrating. Then yeast artificial chromosome. Eukaryotic vectors, maybe have baculovirus, which infects the insect cells. Then we have mammalian based vectors. Today, you people are not at all responding to anything. So, have you studied everything, each and every topic? Because didn't look like when we were discussing questions. Okay, so I will ask questions because you are not uh, responding anything. So I will only ask you questions. Hmm. Tell me the criterias of a good vector and what are the essential components. What are the important components of any basic vector? Tell me. What are the basic components? What all things are needed in a vector? Basic things in a cloning vector. What things are needed in a cloning vector? Hmm? Tell me.
Restriction sites.
Restriction sites, okay. So suppose I'm making this vector map, okay. Low molecular weight, okay, yes, low molecular weight is the criteria for a good vector. So, see, the basic criteria. So, you have a plasmid, and from that plasmid, you design a vector. So, there are certain things that you have to add in this plasmid to make it a vector. The first thing is that a vector should be able to replicate when it is going inside a host cell. So, that means a vector should have. Hmm. Hmm. So, a vector, it should have, first thing is origin of replication. Okay. So that means that any vector, whether it should replicate more or in less, that is a different thing. But at least that vector should be able to replicate inside the host cell. So the basic criteria, the basic requirement for making a vector it is that it should have an origin of replication. Second thing is that it should have a selection marker. That means that there should be some phenotypic differences, there should be some phenotypic changes on the basis of which you can select the vector, you can select your cloned vector. For example, the different types of selection markers that we have studied are the antibiotic selection markers, the blue white screening, or we have studied about the biosynthetic pathways, or in bacteriophage there is plaque formation also. So these are the selection criteria, selection markers that a vector must have. Then the third important thing is that it should have cloning sites. Cloning sites means that it should have, if this is a vector, normally in a vector there is a place which we called as MCS. MCS is multiple cloning sites. So multiple cloning sites means that you have the restriction sites for a wide variety of restriction enzymes. For example, EcoR1, ZO1, BAM-H1, STU1. So that means your vector should have cloning sites where, that means that suppose if you have your insert and you have your vector, so you want to use the restriction enzyme, so you can use the restriction enzymes, only those restriction enzymes you can use whose sites are present in a vector. Suppose if I want to use EcoR1, then EcoR1 site should be present in the vector, only then I can use EcoR1. So therefore, normally in commercial vectors, what is the thing, that in commercial vectors there are a lot of multiple cloning sites, multiple restriction enzyme sites are present already. So you can choose any of the site depending upon your requirement. So MCS means multiple cloning sites. Multiple cloning sites means multiple sites for different kinds of, matlab, sites for different kinds of restriction enzymes. Is it clear? Anybody has a problem? So these are the basic things. So when you were, anybody says that what are the basic things that a vector should have, so if a vector does not have an origin of replication, then how it will replicate? It cannot replicate. When it does not have any selection marker, then how can you screen whether your vector is has uh, taken up the insert or not? So selection marker is needed, and it should have cloning sites, otherwise you won't be able to cut it with any restriction enzyme. Is it clear?
Yes, ma'am.
Hmm. This is the basic criteria of a so Vedangi has said, okay, selectable marker, origin of replication, reason. Yes. So I think now you know the basic criteria of a good vector, of a basic vector. Not a good vector. Good vectors have different things. Now, in that, we can have vectors which can be of prokaryotic origin, and we can have vectors of eukaryotic origin. So, the first ever vector which was made, it was a prokaryotic vector, and that vector was PBR322. So, basically, this PBR322 vector, it is made from three different vectors. Okay. So you can see that the origin of replication is taken from PMB plasmid. The ampicillin resistant gene is taken from RSF plasmid, and the tetracycline resistant gene is taken from PSC101 plasmid. So that means in PBR322 you have selection markers as antibiotic resistant gene. So you have tetracycline resistant gene also, and you have ampicillin resistant gene also. So this PBR322 was the first vector which was made, and this vector was made by taking the few parts of three different vectors. This is a prokaryotic vector. So PBR322 is a prokaryotic vector. So it is a prokaryotic vector, that means it cannot clone eukaryotic genes in it. It can only clone prokaryotic genes in it. Okay. Then the next vector which was made after PBR322, it was made after PBR. But it is a even more modified version of PBR322, which is PUC19. Okay, plasmid, and UC means University of California. This is the full form of PUC. Plasmid, University of California. So the University of California, this vector was made. Now, this vector is even smaller in size than the PBR322. The PBR322 is approximately of how much? 4 kb something. Mm, so so PBR 322 is approximately of 4 kb, whereas this puck is even lesser molecular weight of 2.8 kb. So we have studied also in the criteria of a good vector that a good vector should be of low molecular weight. So puck 19 is a more advanced version of is a more advanced vector as compared to PBR 322, because A, it is of low molecular weight. Your PBR 322 is 4 kb and your puck vector is approximately 2.8 kb. And also it has selection criteria as blue white screening, that is lag z gene selection criteria it has. Okay, so these are the two prokaryotic vectors that we have studied. Then we have studied about some eukaryotic vectors. So if you know, there are n number of vectors which are available in the market now. So which vector you have to use, it depends upon your requirement. So it is not possible to cover each and every vector. So therefore we have covered in our curriculum the vectors that are the most important or the most basic and most studied one. So PBR and PUC are the most studied vectors basically when we talk about the prokaryotic vectors. When we talk about the eukaryotic vectors, so eukaryotes vector can be your yeast vector, phage vector, and mammalian based vectors. Like we have studied that your yeast vectors, there are many different kinds of yeast vectors. Episomal vector, so episomal vectors will produce transient or stable. Tell me, episomal vector will produce a transient or a stable? Transient or stable expression?
Ma'am, transient.
Yes, episomal will produce transient because episomal vector is not able to integrate with the host genome. So if this is your host cell and this is the host DNA, so episomal vector will be present like this as an episome. Whereas an integration vector, it will integrate with the host genome. So it will produce a stable expression. So integration vectors will do a stable expression. Then we have yeast artificial chromosome. Now, yeast artificial chromosome, what is the important criteria apart from the tryptophan and uracil biosynthetic this thing? What is a very unique feature of yeast artificial chromosome? Why it is so much useful? Hmm? Because because it can clone very large DNA fragments of approximately 100 kb. So 100 kb DNA fragments can be cloned in the yeast artificial chromosome. So I think in the last week class, there was a table where they have shown the carrying capacity of different vectors. So yeast yak has the most carrying capacity and plasmids had the least carrying capacity. That means yeast artificial chromosomes can clone a very large DNA fragments, whereas your this thing plasmids can clone very small DNA fragments. Okay, so this is a very important property of yeast artificial chromosomes, that they can clone a very large DNA fragments. Then baculovirus vectors, basically they infect the insect cells. Then you have mammalian vectors. So mammalian vectors such as SV40 vector is important. Then you have bacteriophage lambda based vectors where you have the cos sites at the end. See, these are the cos sites which are present. Left side cohesive ends and right side cohesive ends. These are the cos sites which causes the circularization of the DNA of this lambda DNA, lambda genome. Okay, so with this we have completed this today's lecture. So for the next class, I would really appreciate if you try to at least go through once or twice the lectures, because this time I could not find, most of the people have not read it properly. Also, I have told you in the last class that it would be better if you could read about the primer designing, but none of you asked me. Has anybody tried primer designing in the last week? Nobody has tried now. I told you that you try to design a primer, and then if you have any problems then we can discuss. See, these are the things like setting up a digestion reaction, setting up a ligation reaction, primer designing. These are things that you won't get in books. I mean, theoretically it would be there, but if you want to have some hands on experience, this is the best platform. I think you can have some hands on experience on these things. Otherwise, I don't know how many of you want to pursue for higher research, but if you want to go for higher research, then you have to design primers, and then it would be easier for you if you have the some basic knowledge about it. So anyways, it depends upon you how much you want to utilize the platform and this particular opportunity. So for the next class, I would highly appreciate, again, I'm saying, please read whatever is being taught in the last class, so that you will also not feel bored, and I will also have some more enthusiasm in teaching. Okay? So, will you guys read properly in the next class?
Yes, ma'am.
Okay,
Yes, ma'am.
please read in the properly in the next class. And and I will upload the this thing PPT in my YouTube channel. I think by now you all have the link of it. So you can go and you can, if you have any problem, if you have any queries, you can comment in the comment section, or we can discuss in the next class also. Fine?
Yes, ma'am.
Hmm. Okay, chalo then. Bye bye for now, and happy Independence Day again to everybody.